Okay, regulation of blood sugar. About 30 loves to focus in on this, and I've given you a little workbook assignment to do, which is really important that you do this one. As with all the assignments I give you, I pare them down just to give you the basic key ones. 13.24 has these diagrams in there. Key terms: pancreas, islets of Langerhans, beta cells, alpha cells, insulin, glucagon, diabetes mellitus, hyperglycemia, type 1, type 2 diabetes, and we're going to start with the pancreas. Okay, now Biome 20, you learned that the pancreas was an organ of digestion. It releases bicarbonate ions to buffer the acidic contents of the stomach. It releases enzymes like pancreatic amylase, lipase, some protein digesting enzymes as well. Okay, but a very small percentage of these cells in the pancreas, here we go, very small, called the islets of Langerhans, these islands of cells have two different types of hormone producing cells. So the pancreas is also an endocrine gland. So it's both exocrine, there's a duct to release enzymes and bicarbonate into the pancreatic duct, into the digestive system, and endocrine, where these are going to get released directly into the blood. Endocrine cells, islets, the beta cells secrete insulin, and insulin lowers blood glucose levels. Alpha secretes glucagon, and that raises blood glucose levels. Okay, now this diagram is in your textbook. Now the homeostasis feedback, again, we want to see this kind of sinusoidal graph here. And last time we were looking at calcium levels, calcitonin, if its levels are too high, PTH if it's too low. Now we're going to look at blood glucose levels. We want to maintain homeostasis. If the levels are too high, insulin gets released. If the levels in your blood glucose are too low, glucagon gets released to maintain your BGL levels. Okay, so after a meal, BGL levels are high. Insulin is secreted. Target cells are muscles and liver, become permeable to glucose. Now, your reading on the big assignment also talks about type 2 diabetes and the effect on the nerve cells, neurons in the brain. So take a close reading on that. So your target cells, muscle, liver, become permeable to glucose. It unlocks glucose so glucose can enter these cells, muscles and liver, and muscles and fat tissues and liver. There we go. And blood glucose levels decreases. There they are, they go down. Well, that's going to have a negative feedback on insulin. Insulin's going to go down. But if the glucose levels are too low, then glucagon is going to get secreted. And it's going to convert glycogen into glucose. And glycogen is stored in your liver, okay? And the fat is also going to get, fat cells are also going to get converted into glucose from your fat cells. And this is going to cause the levels, again, in one graph to rise. So interesting Canadian story in this was Banting and Best. Now, he was skeletal, listless, and close to dying in a Toronto hospital bed. 14-year-old Leonard Thompson had type 1 diabetes and timing was running out. There was no cure or effective treatment for then fatal wasting disease that restricts the body's ability to produce or use insulin. But Thompson was about to be entered into medical history books. On January 11, 1922, the boy was injected with a pancreatic extract prepared by Dr. Banting and his medical student, Charles Best. Banting and Best successfully prolonged the lives of diabetic dogs in the University of Toronto. Okay, using extract they prepared. It was originally derived from the dog pancreas, but later sourced from slaughterhouse cattle, which were more plentiful. Before injecting Thompson, they tested the safety of the extract, which they called islatin. Now, take a look at this girl here. Yeah, this was taken about around the same time. Here's a girl who is wasting away, this wasting disease, wasting away, wasting disease, okay? With dog or cattle insulin, all of a sudden, poof, the cells are able to absorb glucose, okay? And gets converted into fat, and child survives. So, effects of insulin therapy in 1922, in a case described, show a young girl insulin deficient before treatment and after. So, let's get this clear. Diabetes mellitus, inability to produce insulin. Symptoms are hyperglycemia, high glucose. Fatigue, fat, and protein is broken down instead of because glucose cannot be used. High blood glucose levels in the urine because it's not being absorbed into target tissues. Thirsty, low energy. Type 1 diabetes is insulin dependent. The immune system, this is autoimmune, attacks the beta cells and requires daily insulin injections. Type 2 is adult onset, non-insulin dependent. Cell receptors stop responding to insulin, develops gradually, and obesity is a risk factor. When your body is being saturated with glucose, the insulin receptors become um, um, non-receptive, okay, to insulin because the body is continually being flooded with food. Insulin is continually being released, and you no longer sensitive. So the best way is through diet and exercise, okay, and then it resensitizes insulin receptors. Okay, now um, this is the last thing I want you to do. You've got a graph. I want you to try page 458 and get this graphed, and then we're going to have something to talk about once you've got this done. Okay. Incidentally, diabetes insipidus is pituitary gland does not make ADH. The kidneys make a lot of urine. Okay, very dilute urine. You might have remembered that lab we did in Bio 20. Okay, here is a graphic organizer. Spend a bit of time going through that. And I put a keyword summary because there's so much here. Some key concepts that you need to know in terms. And then here are some hormones in green. Here are some diseases that we've gone through and effects on them, okay, and the related hormones, okay? So go through that one last time. Read through these, be able to do these, answer these questions, okay? And I'm not going to get you to hand these in. I just want you to be able to know those.